All right, get ready for an absolutely incredible journey. We're going on a tour of one of the most amazing structures in the universe, the eukaryotic cell, and I want you to forget any idea of a simple blob of jelly. We are about to explore a bustling, microscopic city, a metropolis that is the fundamental building block of all complex life. Yep, that includes you and me.
So let me ask you a question. What could a massive sequoia tree, a tiny little swimming amoeba, and, well, you possibly have in common? It seems like a weird riddle, right? Well, the answer is pretty profound. It's the type of cell you're all made of, a super sophisticated, highly organized structure. You're all eukaryotes.
So here's our game plan, our itinerary for this tour. First, we're going to get a bird's eye view of the city. Then, we'll check out its outer walls. After that, we need to get cleared by the smart gate, border control. Then we'll visit the industrial hub, where all the action happens. And finally, we'll zoom out and see how this whole incredible city just works.
Okay, first stop. Let's get that big picture view. We're looking at the two main types of cellular cities you'll find out there, the plant cell and the animal cell. Now, they share a lot of the same basic infrastructure, but as you're about to see, they've also got some really distinct architectural styles. And you can see those differences right here, side by side. Look at the plant cell on the left. It's kind of rigid and boxy, right? That's thanks to its tough outer cell wall. And notice that huge thing in the middle? That's the central vacuole. Oh, and see those little green discs? Those are chloroplasts. They're basically tiny solar power plants. Now compare that to the animal cell on the right. It's much more flexible. It doesn't have a cell wall or chloroplasts, but it has these unique structures called centrioles, which are super important for when the cell needs to divide. Let's zoom in a bit right to the city's perimeter. We're going to check out its first line of defense. And for plant cells, that is one formidable structure, the cell wall.
Now, this is not just some simple fence. It is a critical piece of infrastructure. It gives the cell its definite shape, it protects it from getting crushed or infected, and it even plays a huge role in how it communicates with its neighbors. Think of it as the city's mighty outer rampart.
And this wall is way more complex than you'd think. The materials actually change depending on the organism. In plants, it's this matrix of cellulose and proteins. And see how neighboring cells are stuck together? That's a layer called the middle lamella. It's like cellular cement. But here's the coolest part. These walls aren't solid. They've got these tiny little gates called plasmodesmata. They're like secret tunnels connecting one cell to the next for communication and transport.
All right, now we're moving just inside that wall to a structure that every single cell has, the plasma membrane. If the cell wall is the big outer fortification, this is the city's highly intelligent, super selective border control. It's the ultimate gatekeeper, deciding exactly what gets in and what gets out. So how is this gatekeeper built? Well, our best picture of it is called the fluid mosaic model. The fluid part refers to that main layer of lipids, which kind of behaves like a liquid. And the mosaic part? That refers to all the different proteins that are embedded in it, floating around like tiles in a mosaic. You can see some just sit on the surface, while others go all the way through. So how does this amazing barrier even form in the first place? It's all because of this double layer of molecules called phospholipids. It's such an elegant design. Each one has a water-loving head that faces the watery environment outside and inside the cell, and then it has two water-fearing tails that hide away from the water tucked in the middle. This clever arrangement just naturally creates this stable and really effective barrier.
And to put some actual numbers on this, the ratio of proteins to lipids can really vary. For instance, if you look at the membrane of a human red blood cell, you'll find it's about 52% protein and 40% lipids. So what does that tell you? It tells you that those proteins aren't just for show. They're doing some serious heavy lifting, handling all that transport and communication.
And how does this border control actually work? Well, it uses two main methods. First, there's passive transport. This is the easy stuff. It's like rolling a ball downhill. Molecules just move from a high concentration to a low one. No energy needed. But then there's active transport. For this, the cell has to spend energy, the stuff called ATP, to literally pump molecules against their natural flow. It's like trying to push a giant boulder uphill. The famous sodium-potassium pump is a perfect example of this hard work.
All right, our passports are stamped, we've cleared customs, we are now inside the city's bustling industrial and logistics hub, the endomembrane system. This is where the magic happens. It's where the cell's most important products are built, packaged, and shipped out. So this whole system is really a network of membranes and organelles all working together in perfect harmony.
The main players you need to know are the endoplasmic reticulum, we just call it the ER, and the Golgi apparatus. The best way to think of them is like the city's giant factory complex and its central post office, working hand in hand.
Now, this ER factory comes in two models. First, you've got the rough ER. It's called rough because it's studded with these tiny little structures called ribosomes, giving it a bumpy look. This is the assembly line for proteins. Then you have the smooth ER. No ribosomes here, so it's smooth. Its specialty is making lipids and, in animal cells, things like steroid hormones. Different factories for different products. Makes sense, right? So after things are made in the ER, they get sent over to the Golgi apparatus. And this thing functions exactly like a post office. It's amazing. First, materials arrive in little transport bubbles at the receive side called the sysface. Then, they travel through these stacks, where they get modified and sorted. Finally, they're all packaged up into new bubbles and shipped out from the shipping side, the transface, to their final destination. Now, one of the special packages shipped out by the Golgi becomes the city's recycling and waste disposal center, the lysosome. These are basically little sacks filled with super powerful digestive enzymes. They can break down carbs, proteins, lipids, you name it. They're the cleanup crew dealing with any cellular junk or unwanted materials. Our final stop in this industrial hub is the vacuole, the cell's giant warehouse and reservoir. It's this membrane-bound sac that holds water, sap, and even waste products. And in plant cells, this thing is enormous. I mean, it can take up to 90% of the entire cell's volume. But in other organisms, they have specialized roles. Like in an amoeba, a special contractile vacuole is constantly pumping out excess water to keep the cell from bursting. And that pretty much wraps up our tour of the cell's outer defenses and its industrial heartland. So after seeing all these different departments in action, let's just take one final step back and quickly recap how this incredible little city is organized. Here it is, the perfect cheat sheet for our tour. It just lays it all out. We saw the cell wall as the strong city wall, the cell membrane as the smart border control, the ER as the factory, the Golgi as the post office, lysosomes as the recycling center, and the vacuole as the warehouse. See? Each part has a really distinct job, but they all have to work together in this one beautiful, coordinated system. And I'll just leave you with this final, kind of mind-blowing thought. This intricate city, with all of its complex machinery and perfectly coordinated functions, it is repeated trillions of times just to make you who you are. It's just this powerful reminder of how, in biology, this breathtaking complexity can emerge from the simple interaction of simple molecules. How does that even happen? Honestly, that's a question that continues to drive science forward every single day.